Hello students, in this session of chapter number 3, that is inheritance and variation, we are going to learn the topics deviations from Mendel's findings, incomplete dominance, codominance, multiple alleles and Cleotropy, or it is also called as pleiotropism. First, we will study the topic deviations from Mendel's finding. Due to Mendel's experiment on garden pea plant, few generalizations arise. That is, single trait is controlled by single gene and it shows two alleles. The two alleles then show interaction or interact with each other in which one is completely dominant over the other. According to Mendel, factors or now these factors are called as genes for different trait present on different chromosome assert or express themselves independently. After Mendel, number of deviations or differences were found in post-Mendelian era. Post-Mendelian era means different experiments were carried on genetics by different scientists, and from this they developed their different inventions as well as readings in the field of genetics. So these deviations or differences were found due to additional studies made on genetics by different scientists, which give additional information on the pattern of inheritance of genes. So these deviations or differences in the genetics after Mendel are described as neo-Mendelism. The phenotypic expression of gene. Phenotypic expression means due to presence of a particular gene, organism develop external appearance. It is called as phenotypic expression. So this phenotypic expression of a gene can be influenced or modified by other gene. It is of two types. The first one is intragenic interaction. Intragenic interaction means it occurs between different alleles of the same gene. Example, dominance and codominance. Now you know that when there is a cross between individuals differing in one pair of contrasting character, the character which appears in F1 generation is called as dominant and the another one is called as recessive. So this is also called as law of dominance. Then in codominance, both the genes or alleles of an allelomorphic pair express themselves equally in F1 generation, and these individuals are called as F1 hybrids. Then it occurs between multiple series of genes. The second phenotypic expression is intergenic, or it is also called as non-allelic. Intergenic means non-allelic interactions occur between the alleles of different genes present on the same or different chromosomes. Example of it is Cleotropy or polygenes. Cleotropy means a single gene controls two or more different traits. Then polygenic trait is one whose phenotype is influenced by more than one gene. Now we will study the law of incomplete dominance. In incomplete dominance, both the alleles or alleles are also called as genes, of an allelomorphic pair express themselves partially or incompletely. One gene cannot suppress the expression of the other genes or alleles completely. Hence, there is incomplete expression. Then, in such cases, there is intermediate expression of both the traits in F1 generation plants, which are called as F1 hybrids. Example of it is a plant called as Mirabilis jalapa. Mirabilis jalapa in Marathi is called as Gulbakshi. So, in Mirabilis jalapa, red flowered plant with the genotype capital R is crossed with white flowered plant with the genotypes small r. Then the F1 offsprings which are produced have pink flowers with genotype that is capital R and small r. Here we see a cross between a plant with red flowers with a plant with white flowers. A red flowered plant shows genotype that is capital R, while white flowered plant shows genotype small r. So when these plants are crossed, equal number of gametes, that is male and female gametes, are produced and they unite and form F1 generation plants which are called as hybrids. So the plants which are produced in F1 generation produce pink flowers. So here we see that one gene cannot suppress the effect of other genes. So both the genes or both the alleles express themselves partially and produce a intermediate color between red and white, that is pink flowers. When the plants bearing pink flowers are allowed to self-pollinate or artificial self-pollination is carried out, it is called as selfing of F1 hybrids. Then we get F2 generation. So in this generation, equal number of female gametes and equal number of male gametes are produced and we get a different combination. So here we see capital R, then capital R. We see red flowered plants are produced. Then here capital R, small r, pink flowered plants are produced. Then small r, capital R. Here we see pink flowered plants are produced. And both the R, that is small r, white flowered plants are produced. So here we see that the plants produced, that is plant with red flower having genotype, that is both capital R, is in the ratio of 1. Then plant with pink flower having genotype capital R small r in the ratio of 2, and plant with white flower having genotype, that is small r is produced in the ratio of 1. So here we see it is genotypic ratio. Then phenotypic ratio is similar or is same to that of the genotypic ratio, that is 1 red, 2 pink and 1 white flowered plant are produced. So from this we study that when two individuals are crossed, then both alleles of an allelomorphic pair express themselves partially. One gene cannot suppress the effect of the other genes. Hence, they are incompletely expressed. Hence, the law is called as law of incomplete dominance. Now, we will study the law of codominance. In codominance, both the alleles, or they are also called as genes, of an allelomorphic pair express themselves equally in F1 hybrids. Such alleles which express themselves equally and independently in F1 hybrids are called as codominant alleles. Hence, in codominance, both alleles are expressed. For example, when there is a cross between red coat colored cattle or red haired cattle with the genotype that is R and R crossed with a white colored or white coat cattle with the genotype WW. Then in F1 hybrid, the cattle or individuals produced are roan colored with a genotype that is R and W. Roan colored means roans are the mixture of red and white color here. So in codominance, both the traits are expressed equally. Here we see a cross between red colored female with a cross between white colored male. Here we see the phenotype. Phenotype, you know that phenotype is the external appearance. So externally, one is red and one is white. That is a red female and white male with the genotype, or they are having homozygous genotype. Here we see that red colored female shows both R and white color male shows both W. Hence they are called as homozygous. So when they are crossed, they produced equal number of male and female gamete. And in F1 generation, we see all the cattle produced are having roan color. Roan color, you know that roan means a mixture of red and white strips. So the F1 individuals, that is roan colored female, is crossed with roan colored male. Equal number of gametes, here we see equal number of male and female gametes are produced. And then in F2 generation, we see that here a red colored cattle is produced and it is female having genotype, or it is homozygous, both are R. Then a roan colored female with genotype RW is produced. Then another roan colored male with genotype RW is produced. So here one red colored, two roan colored, and here we see white colored male is produced. So the ratio is, now you know that the phenotypic ratio or external, how the individuals appear. So phenotypic ratio is one red, then two roan and one white. So 1 as to 2 as to 1. Similarly, the genotypic ratio is the same. Here we see homozygous R, then in the ratio it is 1. Then roan colored, that is genotype is RW, is in the ratio of 2, and white color is in the ratio of 1. So both phenotypic and genotypic ratio are one and the same. So in codominance, both the alleles or genes of an allelomorphic pair express themselves equally in hybrids. And in F2 generation, we see the phenotypic and genotypic ratio, that is 1 as to 2 as to 1. So in codominance, when there is a cross between red and white cattle, both the genes express equally. Hence, the law is called as law of codominance. Now we will study multiple alleles. More than two alternative forms or alleles of gene in a population occupying the same locus on a chromosome or its homologue are called multiple alleles. Multiple alleles arise by mutations of wild type of gene. A gene can mutate several times or many times, producing alternative or different expressions. Now you know that mutation means that there is a sudden change in the genetic makeup of an individual. Due to this mutation or change in genetic makeup, an individual or organism develops different abnormal characters. In Drosophila, multiple alleles are found. Different alleles in a series show dominant recessive relation or may show codominance or incomplete dominance among themselves, while wild type is dominant over the other mutant alleles. In Drosophila fly, large number of multiple alleles are found. Here in the diagram, we see homozygous genes are responsible for the different types of wings. In Drosophila, due to the mutation, different types of wings are found due to the multiple alleles. First, or in the first type, we see normal wings. These normal wings are due to the genes VG plus. So, generally, in normal Drosophila fly, the wings are also normal as they are not mutant. These are due to the genes VG plus. Then next we see nicked wings. Nicked wings means they are reduced due to the genes, that is VGNI. Then next we see notched wings. Notch means there are incisions on wings. These notched wings are developed due to the genes VGNO, that is notch in the wing is developed. Then in the next we see the wings are like strip, hence it is called as strap wings. And these strip like or strap wings are produced due to the genes VGST. Then at the last we see vestigial wings. Vestigial wings means wings are not developed in Drosophila due to the gene, that is VG. So mutations are developed in Drosophila due to multiple alleles arised by mutation. Then another example of multiple alleles is blood group in human being. That is, we see blood group A, B and O These are present due to these multiple alleles. Now we will study the topic, that is pleiotropy. When single gene controls two or more traits is called pleiotropy or pleiotropism, and the gene is called as pleiotropic gene. The disease sickle cell anemia is caused by the gene, that is HBS, in human being, and normal healthy gene, that is HBA, is dominant. The carriers, which are heterozygotes having genotype, that is HBA HBS, show signs of mild anemia as their RBCs become sickle shaped, or they are also called as half moon shaped. Abnormally, due to low concentration of oxygen. Then the homozygotes with recessive gene, that is HBS, die due to sickle cell anemia because of very low concentration of oxygen. So thus gene for sickle cell anemia is lethal, or it is also called as dangerous in homozygous condition, that is HBS, and it produces sickle cell trait or sickle cell character in homozygous carrier with two different expressions that are produced by a single gene. Here in the diagram we can see the results when there is a marriage between two sickle cell carrier individuals. Here we see the parental generation having phenotype. Both the phenotype is sickle cell carrier. Then their genotype is HBA HBS. Here we see HBA HBS. When these two individuals get married, then the offspring produced show the ratio, that is 1 as to 2 as to 1. Now here we see the genotype of the offspring is HBA HBA, means it shows both the dominant genes and it is homozygous. Then here we see the individual or offspring with genotype, that is HBA HBS, and it is in heterozygous condition. It is a sickle cell carrier. Here also we see HBA HBS. It is also one of the sickle cell carrier. And lastly, we see both the genes are similar, which cause the disease sickle cell anemia, that is HBS HBS. So here we get the ratio, that is normal offspring having dominant genotype in the ratio of 1. Then we get two sickle cell carriers which show mild signs of anemia. And lastly, we see that the lethal gene is present in homozygous condition. So this individual having the genotype HBS HBS die due to the disease sickle cell anemia. Sickle cell anemia means there is deficiency of oxygen in RBCs, due to which the individual dies. So till now in this chapter we have completed chromosomes and mechanism of inheritance, reasons for Mendel's success. Then we have studied different terms used in genetics. Then we have studied monohybrid cross, dihybrid cross, back cross, and test cross. Similarly, we have studied the different laws of Mendel, that is law of dominance, law of segregation, then law of independent assortment. Then we have also studied deviations in Mendel's finding, in which we have learned incomplete dominance, codominance, multiple alleles, and Cleotropy. So we have completed all the topics